Bovines, such as cows and bulls, inhabit a sensory universe that shapes their perception of the world around them. Acquiring knowledge of the complexity of their senses offers priceless insight into their structure, components, and functions. This presentation aims to illuminate the amazing capacity of these animals to survive and navigate a wide range of situations. So now we will be looking at the classification of the sense organs of mammals. Firstly, the eyes, and then the ears, followed by the nose, and also the tongue. Finally, the skin.
The eyes are the sensory organs that allow an organism to perceive visual information. It is made up of the sclera, cornea, iris, pupil, lens, retina, tapetum lucidum, and optic nerve. These structures and components enable the eyes to detect and capture light, then convert it into nerve impulses for the brain to interpret. The sclera is the outer layer of the eyeball that extends from the cornea to the optic nerve. It's thin and white due to an abundance of randomly interwoven collagen fibers. The sclera is made up of three layers: the episclera, which is the outermost layer, sclera proper, which forms the majority of the sclera, mainly consisting of the dense connective tissue, and lastly lamina fusca, which is the innermost layer. The sclera is important because it's attached to extraocular muscles which function to move the eyes, further allowing the organism to see. Most mammals have 6 extraocular muscles, which are the superior rectus, which allows the eye to elevate, inferior rectus, which allows the eyes to depress, medial rectus, which allows eye adduction, lateral rectus, which allows eye abduction, superior oblique, which helps move the eye downward and inward, and lastly the inferior oblique, which helps to move the eye upward and outward. However, bovines, caprines, and ovines only have 4 out of 6 extraocular muscles, as they do not possess the superior and inferior oblique extraocular muscles. The cornea is a dome-shaped transparent membrane that covers the front part of the eye. It's avascular, has an abundance of nerves, and is made up of dense connective tissue with predominantly type 1 collagen. These type 1 collagen fibers are uniform and parallel to each other in terms of arrangement to allow transparency of the cornea. The cornea consists of 5 layers: the epithelium, Bowman layer, stroma, Descemet's membrane, and corneal endothelium. The epithelium is the outermost layer, while the corneal endothelium is the innermost layer of the cornea. The Bowman layer functions to protect the stroma, which is the largest layer. And lastly, Descemet's membrane serves as modified basal membrane. This allows the cornea to refract light and focus light rays onto the retina. The iris is colored tissue that contains the pupil. Irises can have different colors, however in bovines their iris is brown. The iris is composed of 2 types of tissue: heavily pigmented epithelium, which gives the color of the iris, as well as smooth muscle tissue, which allows the iris to perform its function of controlling the diameter and size of the pupil. There are 2 layers of this smooth muscle tissue. First is the dilator pupillae, which functions in enlarging the pupil. Next is the sphincter pupillae, which functions in constricting the pupil. The pupil refers to an opening or absence of tissue found within the iris. It functions to allow light to enter the eye and reach the retina. So there are 3 main pupil shapes that mammals can inherit. First is the vertical slit. This one is commonly possessed by predators, like cats, that often hunt in low light conditions. Next is circular. The circular shape is commonly possessed by diurnal hunters, like larger cats, like lions and tigers. Lastly is horizontal or oval, which is commonly possessed by prey animals, for example, bovine, ovine, caprine, and equine. The lens is the transparent curved structure located behind the pupil. It is made up of 3 layers: the lens capsule, lens epithelium, and lens fibers. The lens fibers form the transparent structure which allows light to pass through. This is important as light rays that pass through the lens will bend to form clear images at the back of the retina. The retina refers to the light-sensitive layers of nerve tissue located at the back of the eye. It receives light rays and converts them into nerve impulses for the brain to interpret. It also consists of glial cells and photoreceptor cells, also known as rods and cones. Rods are responsible for light detection, while cones are responsible for color vision. Mammals typically have 3 types of cones, which are the cones sensitive to short wavelength or blue light, medium wavelength or green light, and long wavelength or red light. However, not all mammals possess all 3 cones. For example, bovines only have 2, meaning they only have cones that are sensitive to short and medium wavelength light. This results in them having dichromatic vision, where they can only see in hues of blue and green. Connecting to the retina is the optic nerve. The optic nerve is composed of bundles of nerve fibers, primarily axons, that function to relay impulses from the retina to the brain, specifically the visual cortex. Lastly, The tapetum lucidum is a layer of tissue located behind the retina of mammals. Although they are present in mammals, they are actually absent in humans. The tapetum lucidum is a retroreflector, meaning it reflects visible light through the retina. This increases the chances of the photoreceptors in the retina to detect light, hence allowing for increased visual sensitivity under low light conditions, or in other words, night vision.
Let's take the example of the feline ears. Ears is the auditory sense organ of the body. The ears help to perceive sound and function in balance. The ears are divided into three major parts: the outer ear, the middle ear, and the inner ear. The outer ear consists of the external ear flap and the ear canal. The anatomy of the feline pinna includes several key components. Cartilage. The feline pinna is primarily composed of cartilage, a flexible and lightweight connective tissue that gives the pinna its shape and structure. Blood vessel. Blood vessel between the pinna helps regulate temperature by constricting and dilating to conserve or release heat respectively. Nerves. Nerves between the pinna transmit sensory information, allowing cats to detect touch, movement, and changes in their environment. Muscles. The pinna is equipped with small muscles such as the auricular muscles, which controls its movement. These muscles enable the cats to rotate, tilt, and flatten their ear in response to various stimuli. The anatomy of the feline ear canal includes several important structures. Stenoacoustic meatus. It is a tube-like passage that extends from the outer ear to the eardrum. Cartilage. The outer portion of the ear canal is lined with cartilage, which provides structural supports and help maintain the shape of the ear canal. Inner lining of the ear canal is covered with skin, which contains glands that produce cerumen. Ceruminous glands. These are specialized glands located within the skin of the ear canal that produce cerumen. Hair. Some cats may have fine hair lining their ear canals. Nerves. Nerves between the ear canal transmit sensory information, allowing cats to detect touch, pressure, and pain. Moving on to the middle ear. The feline middle ear consists of the tympanic membrane, eustachian tube, and the tympanic cavity. These structures are responsible for transmitting sound vibration from the outer ear to the inner ear. Additionally, the middle ear helps to regulate pressure and maintain equilibrium in feline. First is the eardrum. The feline eardrum, also known as the tympanic membrane, is made primarily of thin layers of tissues composed of collagen fibers and epithelial cells. These layers are stretched across the ear canals and they vibrate in response to sound waves, transmitting this vibration to the middle ear. Next is the ossicles. The feline ossicles, which are small bones located in the middle ear, are primarily made out of bone tissues. They consist of malleus. It is shaped like a hammer and is attached to the inner surface of the tympanic membrane. Incus. It is shaped like an anvil and is situated between the malleus and the stapes. Stapes. It is shaped like a stirrup and attached to the inner ear. These bones are crucial for transmitting and amplifying sound vibration from the tympanic membrane to the inner ear. Next is the eustachian tube. The feline eustachian tube is primarily composed of cartilage and mucous membrane. The cartilage in the feline eustachian tube is primarily composed of hyaline cartilage. Lastly, in the middle ear is the tympanic cavity, also known as the middle ear cavity. It is the space between the middle ear that contains ossicle and is surrounded by the temporal bone of the skull. It is filled with air and plays a crucial role in transmitting sound vibration and maintaining proper pressure balance between the middle ear. Lastly, let's move on to the inner ear of the feline, that consists of several structures that plays vital roles in hearing and balance. First is the cochlea. The feline cochlea is primarily composed of bone and soft tissues. The bony cochlea is a spiral-shaped structure within the inner ear and it contains the sensory organ responsible for hearing. Between the cochlea, there are various soft tissues including the basilar membrane and the organ of Corti, which contains specialized cells responsible for detecting sound vibration and transmitting them to the auditory nerve. Next is the vestibular system. The feline vestibular system is primarily composed of specialized structure within the inner ear, including the vestibule and the semicircular canals. Last but not least is the auditory nerve. The feline auditory nerve, also known as the vestibular cochlear nerve, is primarily composed of nerve fibers. These nerve fibers originate from the hair cells located in the cochlea and the vestibular organs within the inner ear.
Nose are always associated with the sense of smell. Nose has inner lining that contains specialized microscopic structures called olfactory receptors, which provide animals with the sense of smell. Nasal cavity is one of the main structures in nose. Nasal cavity begins at nostrils and ends at choana. It divided longitudinally by nasal septum. It consists of external nasal cavity, which is nostrils, nasal plenum, and philtrum, and internal nasal cavity, which is nasal concha, olfactory epithelium, olfactory bulb, and olfactory tract. Nostrils are nasal openings for entry of air containing odor molecule and vary in size and shape to capture scent based on ecological needs. It consists of basal cells, non-neuronal supporting cells, and olfactory sensory neurons. The function of nostrils is to help passing air from external environment inside the nose. Nasal plenum is pigmented, hairless, rostral-most surface of external nose, while philtrum are mid-sagittal external crease in nasal plenum. Both nasal plenum and philtrum consist of basal cells, sustentacular cells, and olfactory receptor cells. The function of nasal plenum is to help dissolve odor particle in air, while philtrum helps in detecting pheromones and other scent molecules. Nasal turbinates are cartilage which arise from ethmoid bone. It covered with mucous membrane and have rich blood supply. Nasal turbinates are more complex in animals with better sense of smell as they increase the surface area of olfactory receptors. The characteristics are long, narrow, curled shelf bone and shaped like an elongated seashell. It composed of pseudostratified columnar, ciliated respiratory epithelium with thick, vascular, erectile glandular tissue layer. The function of nasal turbinates is to warm and humidify incoming air. Warm and moist air can enhance the ability of odorant molecules to dissolve in mucus and bind to olfactory receptors. Most mammals have three nasal conchae. First, superior nasal conchae is the smallest and located highest in nasal cavity. It connects to middle conchae by nerve endings, serves to protect olfactory bulb. The function is to help protect olfactory bulb and associated with the sense of smell. Second, middle nasal conchae are also part of ethmoid bone. It larger than superior nasal conchae and helps to regulate airflow and important for trapping particles and warming air. Third, inferior nasal conchae, which is the largest and it extends horizontally along lateral wall of nasal cavity in humidifying air and also filtering air. Olfactory epithelium is the specialized tissue that is crucial for sense of smell. It contains olfactory receptor neurons that detect odor molecules. It is found in upper part of nasal cavity next the olfactory cleft of nasal cavity. It is one of the types of pseudostratified columnar epithelium involved in sensing smell. Olfactory epithelium function in detect odor molecules. Olfactory epithelium composed of three main cells. First, basal cells that is essential for regeneration of olfactory epithelium, which occurs every 2 to 4 weeks. Second, supporting cells that provide metabolic and physical support for olfactory sensory neurons. Olfactory sensory neurons are the actual receptor cells that detecting odor molecules. Each neuron has cilia that increase the surface area for capturing odorants. Olfactory bulb is situated at the base of brains. It's a paired structure and contains several types of cells. Olfactory bulb act as main relay station in olfactory pathway. The secondary axons forming olfactory tract. Olfactory bulb function as it receives axon from olfactory receptor cells, which synapse with mirror cells within bulb and process signal before sent to brain. The components of olfactory bulb is mitral cells, tufted relay neurons, granule cells, periglomerular neurons, and olfactory receptor cells. Olfactory tract extends posteriorly from olfactory bulb, running along the underside of frontal lobe. It composed of the axons of mitral and tufted cells from olfactory bulb. The tract divides into medial and lateral olfactory striae. The function of olfactory tract is olfactory tract transmit the process signals from olfactory bulb to primary olfactory cortex and other related area. These regions are involved in identifying odors. When odorant molecules bind to receptor in olfactory epithelium, they trigger electrical signals that travel through olfactory bulb and olfactory tract to the brain, where the smells are identified and processed.
Next, the three main parts of the bovine tongue are root, body, and apex. The root of the tongue is made in cattle or ruminants. The very thick root in the structure of the bovine tongue attached to the hyoid point and mandible. For the body of the bovine tongue, there are three surfaces of the body, which are dorsal, ventral, and ventral. Dorsal surface is slightly routed, ventral surface is related to phenollingual, lateral surface is right and left lateral, flat and routed in structure, apex is thin, flat, and routed. There are four main components in the bovine tongue, which are muscular tissue, mucous membrane, papillate, and salivary glands. Muscular tissue is primarily composed of skeletal muscle fibers arranged in intricate patterns to allow for various movements. The bovine tongue contains intrinsic and extrinsic muscles that facilitate its movement. Intrinsic muscles are responsible for fine movement within the tongue itself, while extrinsic muscles control broader movements such as protrusion, retraction, and lateral movement of the entire tongue. These muscles are crucial for tasks like grasping, manipulating food, and ending in swallowing. The mucous membrane covers the surface of the bovine tongue. It consists of epithelium tissue supported by connective tissue. This membrane plays a vital role in protecting the underlying structure of the tongue, facilitating the movement of food and participating in taste secretion. Papillae has more projection on the dorsal surface of the tongue. They vary in size and shape and are involved in taste perception and manipulating food during mastication. The four main types of papillae found on the bovine tongue are filiform, fungiform, folate, and circumflate. Each type has a specific structure and function related to taste and mechanical process of food. For filiform papillae, they are the most numerous and smaller types of papillae on the bovine tongue. Their cake-like structure distribute across the surface of the tongue, giving it a broad district. Filiform papillae aim in the manipulation of food by providing friction against the structure of the ingest material. However, they do not contain taste buds. For fungiform papillae, they are mushroom-shaped structure interspersed among the filiform papillae. These papillae are larger and more prominent than filiform papillae with a rounded top. They are scattered throughout the dorsal surface of the tongue, particularly concentrated throughout the tips and edge. Fungiform papillae house taste buds, allowing for the perception of different flavors present in food. Next, folate-form papillae are leaf-like structure located on the lateral margins of the posterior part of the tongue. They appear as ridges or folds with numerous small projections. Folate papillae contain taste buds and are involved in taste perception. The last type of papillae is circumflate papillae. They are large, dome-shaped structure located throughout the back of the tongue. They are surrounded by fish-like glue and are the largest papillae on the bovine tongue, typically arranged in a V-shaped row near the base of the tongue. Circumflate papillae are rich in taste buds and play a significant role in taste perception, particularly for bitter taste. Salivary glands secrete saliva into the oral cavity, including the area around the bovine tongue. Saliva plays a crucial role in lubricating food, facilitating swallowing, and initiating the digestive process. The tongue helps distribute saliva throughout the mouth during mastication, ensuring thorough mixing of saliva with injects food for effective digestion.
The skin functions as a sophisticated sensory organ, housing a diverse array of sensory receptors and nerve endings that enable us to perceive touch, pressure, temperature, and pain. The skin comprises three main layers: the epidermis, dermis, and hypodermis. It is also consists of hair follicles, sweat glands, sebaceous glands, and melanocytes.
So now we are going to start with the first layer of the skin. The epidermis is the outermost and thinnest layer of the skin of a bovine. It contains specialized sensory receptors known as dermal cells located in the deepest layer of the epidermis, associated with nerve endings and play a role in detecting light, touch, and pressure. The epidermis is composed of 5 layers of its own: stratum corneum, stratum lucidum, stratum granulosum, stratum spinosum, and stratum basali. And the epidermis functions to protect, regulate water loss, and synthesize of vitamin D. Next is the thick layer of skin located beneath the epidermis, the dermis, providing structural support, elasticity, and various sensory functions. It consists of 2 main layers, the papillary dermis and the reticular dermis, and it houses a variety of sensory receptors, including the Meissner's corpuscles, Pacinian corpuscles, and the Ruffini corpuscles. The papillary dermis is the uppermost layer of the dermis, lying directly beneath the epidermis, composed of loose connective tissue containing collagen and elastic fibers, characterized by finger-like projections called dermal papillae, which extend upwards and interlock with the epidermis, forming the dermal-epidermal junction. Dermal papillae contains capillaries, nerve endings, and touch receptors, or also known as Meissner corpuscles. Meissner corpuscle contributes to the skin's sensitivity to touch. These receptors are sensitive to light touch and low-frequency vibrations. Next is reticular dermis. The deeper and thicker layer of the dermis contributes to the skin's resilience, stability, and overall structural integrity, composed of dense irregular connective tissue containing collagen and elastic fibers arranged in a mesh-like pattern. Sensory receptors for pressure, Pacinian corpuscles, and temperature, Ruffini endings, are present in this layer. Pacinian corpuscles respond to deep pressure and high-frequency vibrations, while Ruffini endings detect stretching of the skin and are involved in proprioception, awareness of the body position.
Now we are on the last layer of the skin, the hypodermis, or also known as the subcutaneous tissue located beneath the dermis. It is composed of adipose tissue, connective tissue, blood vessels, and nerves. Nerves that is densely innervated with sensory nerves that transmit signals from sensory receptors to the brain, allowing the bovine to perceive sensations such as touch, pressure, temperature, and pain. Sensory nerve includes mechanoreceptors respond to mechanical stimuli such as touch, pressure, and vibration. Thermoreceptors detect change in temperature. Nociceptors specialize nerve endings that detect pain.
Now we are on the other components of the skin. First is the hair follicles for regulation of temperature. Next is the sweat glands to regulate body temperature, and followed by sebaceous glands to produce and secrete sebum, lubricates the skin to protect against friction, and lastly the melanocytes acts as an internal sunscreen to defend from ultraviolet damage.
In conclusion, animals have an advanced variety of sensory organs that allow them to successfully transfer their surroundings. Their sense of smell helps them to communicate and locate their food sources. Individual recognition, movement detection, and depth of perception are all made possible by their visual system. The ability of an animal to hear sound waves aid in communication and predator avoidance. And also, their tongue help them to taste food, access food quality, as well as make nutritional decisions. And finally, the sense of touch, which is provided by the skin, promotes social bonding and exploration. Collectively, these sensory organs highlight how adaptive and resilient these animals are in their environment, emphasizing the need to appreciate, recognize, and understand their sensory capacities.